this is the cell cycle. G1 is the period of cell growth before DNA is duplicated, and that is where the interphase begins. S phase is the period when DNA is duplicated. G2, a period after DNA is duplicated, cell prepares for division, and this is where the interphase ends. The next part of the cell cycle is mitosis, which we will talk more in depth about later. Prophase is the first part of mitosis, then is followed by metaphase, anaphase, and telophase. The last part of the cell cycle is cytoplasm division, which is called cytokinesis, which is not to be confused with part of mitosis. In mitosis, the nucleus divides to produce two nuclei that are genetically identical to each other and the parent nucleus. To prepare for division, the DNA replicates in the preceding interphase. Although the chromosomes are not yet compact and visible as discrete bodies, we illustrate them as such to show the formation of the second chromatid per chromosome. Also during interphase, the cell replicates its centrosome. Centrosomes are microtubule organizing centers, which are very important for chromosome movement during mitosis. In many organisms, each centrosome contains a pair of centrioles. In the first phase of mitosis, called prophase, chromatin coils and condenses, resulting in compacted chromosomes that are visible with a light microscope. In prometaphase, the nuclear envelope breaks down and microtubules emanating from the centromeres attach to the chromosomes. The microtubules attach to the structures called kinetochores, which are found in the centromeres of the chromosomes. In metaphase, the centromere regions connecting paired chromatids become aligned in a plate at the cell's equator. In anaphase, the centromere pairs from each chromosome separate, and the single chromatids, which are now considered individual chromosomes, move toward the poles. In telophase, the separating chromosomes reach the poles, and the division of the cytoplasm, called cytokinesis, also begins during this phase. As the cells enter the next interphase, the nuclear envelopes and the nuclei reform, and the chromatin again becomes diffuse. The rate and timing of cell division in your body normally are very precisely regulated. Cells are formed, mature, and eventually die. As this happens, new cells divide, creating replacement cells. Chemical messengers that pass between neighboring cells can help keep the rate of cell division equal to the rate of cell death. And begins to flow to its own pattern of cell division. This precancerous cell divides more often than normal, eventually producing a mass of cells that also divide more often. Further changes in these cells can increase the frequency of cell division even more until eventually a cancerous tumor develops. At this point, the tumor grows large, but it is confined to the tissue where it originated. Late in the development of cancer, some cells may gain the ability to move into blood vessels and travel to other parts of the body. Once established in new locations, these cells continue to divide in an uncontrolled fashion, eventually producing new tumors. Before meiosis begins in the preceding interphase, DNA replication takes place. At this point, the chromosomes are not condensed and are not visible under a light microscope. However, we depict them in a condensed manner so that you can see the cell replicate each chromosome to form two chromatids. Meiosis consists of two cell divisions, meiosis I and II. As the diploid cell enters the first phase of meiosis I, called prophase I, its chromatin condenses into discrete chromosomes. The cell has two centrosomes, which form the mitotic spindle as the centrosomes migrate to opposite poles of the cell. This diploid cell contains homologous chromosomes, which pair up later in prophase I. One chromosome in each pair is maternally derived, indicated by blue, and one is paternally derived, indicated by red. The pairing is called synapsis. At this time as well, the nuclear envelope breaks down. Late in prophase I, the homologous chromosomes exchange genetic material as indicated by crossing over points called chiasmata. Recombinant chromatids consist of both maternally and paternally derived DNA. In this phase, microtubules attach to the chromosomes, directing them to the equatorial plate. At metaphase I, homologous chromosomes have lined up on the equatorial plate in a pairwise fashion, with one homologue on either side of the plate. Note that the chromosomes assort independently. For example, the maternal chromosomes align randomly, not necessarily on the same side of the plate. During anaphase I, chromosomes from each pair move to opposite poles of the cell. The centromeres of the chromosomes do not divide, so each chromosome still consists of two sister chromatids, which now may not be genetically identical due to crossing over. In some species, telophase I occurs in a stage which chromosomes decondense, nuclear membranes reform, and cytoplasm divides in a process called cytokinesis. A short interphase period called interkinesis may then follow telophase I. Note that DNA replication does not take place during interkinesis. During the second half of meiosis, in prophase II, the chromatin again condenses into discrete chromosomes. There are now only a haploid number of chromosomes per cell. Each chromosome consists of two chromatids joined together by centromere. At metaphase II, the chromosomes have lined up on the equatorial plate. Microtubules from opposite poles attach to each sister chromatid of a chromosome. During anaphase II, the centromeres divide and both chromatids become independent chromosomes and move to opposite poles of the cell. During telophase II, chromosomes again decondense and nuclear membranes reform. Depending on other species, cytokinesis may occur.